9.1 DNA technology. DNA technology and recombinant technology. Then in 1.4D, DNA profiling. This is also DNA fingerprinting originally. Then in 12th chapter, DNA technology in disease and medicine. DNA technology in disease and medicine. So many times they mentioned that DNA technology, recombinant technology, both are same. This DNA profiling also almost same. This DNA technology in medicine and diseases. This is dimension, but same. Otherwise, genetic engineering or gene manipulation. These are all same. Okay. What is DNA technology? Genetic manipulation or genetic engineering is known as DNA technology. Gene manipulation or genetic manipulation or genetic engineering is known as DNA technology. See, what is DNA technology? Is a specially designed technique to achieve some targeted goals, specified goals by manipulating genes, that is DNA. What is the definition for DNA technology? That is, first of all, manipulation of genes or genetic engineering is known as DNA technology. So, DNA technology is a specially designed technique. A specially designed technique. Useful to achieve targeted goals. Useful to achieve targeted goals by manipulating genes. That is DNA. Gene also DNA, no? So that is DNA technology definition. That means DNA technology is nothing but genetic manipulation. It is an engineering of genetics. So DNA technology is a specially designed technique useful to achieve specified goals by manipulating the genes. So this is definition. Yeah. The process of DNA technology. What is the process of DNA technology? Yeah. See, what is that? Golden rice. What is a golden rice? Golden rice. What is BT cotton? Genetically modified cotton crop. Is it? Yeah. What is golden rice? That is also same, genetically modified rice variety. See, this is technology. Technology is an application of science. Science is a fact, but technology is an application of scientific facts. Is it? So in nature, some bacteria are there, particularly known as Bacillus thuringiensis, BT. That bacteria produce some toxic substances, endotoxin, exotoxin. So that endotoxin is harmful to only insects. There is no way harmful to animal, at the same time human. So this is the fact. On the basis of that, scientists extracted that endotoxin producing gene and finally inserted into the cotton. Then they produced BT cotton. That means BT cotton is a pest resistant cotton variety. Is it? Pest means which destroy. Is it? Yeah. So particularly, what is that pest? Insects. The insect which spoils the cotton fruit. The cotton fruit is known as bolls. Boll. Is it? To protect the boll and to kill that worms, we introduced some genetically modified variety. That is BT cotton. That's why BT cotton also known as boll gourd. Is it? You know this term? Boll gourd. Boll gourd. Cotton variety. Something like that. What is this golden rice? It is also genetically modified rice variety. How they modified that? See, throughout the world, the major crop, staple crop, there is a rice. Majority people consume that throughout the world. At the same time, this rice has no vitamin A. Vitamin A is totally absent. At the same time, throughout the world, vitamin A deficiency is very high. Due to that, number of individuals are getting blindness. That is a deficiency of vitamin A is a major reason for the increasing of blindness throughout the world. So they extracted that vitamin A producing gene from the carrot, then finally inserted into rice variety. Then they produced vitamin A rich rice variety. Vitamin A scientific name carotenol. That's why what is golden rice? So carotenol rich rice variety. Producing of such genetically modified plants or animals or insects or fowls, fishes, that technology is known as DNA technology. Yeah. So scientists inserted growth hormone into animal, fowls, fishes. Now they produced maximum the rat, total muzzle, maybe mouse, small rat, 20 grams 25 grams maximum. After inserting the growth hormone gene, they produced almost 4 kilo weight mouses, rats. In Western countries, they are eating now. They are becoming major source of the food now. Even rats also, of course. Like that, genetically modified animals, sheep, goats, cows, buffaloes, number of they produced. Such animals are known as transgenic animals, such as genetically modified plants, BT cotton, golden rice, MON 369, maize variety, that one. These are all transgenic plants. That means DNA technology is useful to produce genetically modified crops, genetically modified animals, and even number of medical substances, therapeutic substances, drugs. How we are producing insulin? Insulin is available in the market. Is it? From where they extracted that insulin? That insulin producing gene extracted from the human, then finally inserted into the microorganism E. coli, then they are producing abundant quantity of insulin. Then after purification, they are sending to the market. Yeah. That means we are using microorganisms, animals, plants, fishes, fowls to produce abundant number of genetically modified species. That technology which involve in the production of genetically modified plants, animals, microorganisms, that is known as DNA technology. Okay. That means, what is this DNA technology? Manipulation of genes. This is a genetic engineering process. That's why this is also known as genetic engineering or genetic manipulation or something. Yeah. It's a specially designed technique. Yeah. It's a process. Technical number of techniques apply. That means specially designed technique useful to achieve targeted goals. We want something. We want to produce insulin. We want to produce growth hormone. We want to produce any hormone or enzyme or vitamin or any medical substance, or we want to produce high yielding varieties, yes, pest control varieties, drought resistant varieties. Is it? Even in polar regions also we are producing rice. Deserts also we are producing rice. So how? By using this DNA technology. That means in deserts, drought resistant varieties are there. In polar region, cool resistant varieties are there. In normal region also, they consume less water, less mineral, but they produce high, that means carbohydrate rich, protein rich, or any something. So producing such a targeted plants, targeted animals, that is known as DNA technology. That is introduction. I hope it's clear. Is it? What is this? Then, what is the procedure involved here? See, very simple. First, we should extract the desirable gene. BT cotton. Okay, no problem. So, that endotoxin producing gene we have to extract first. Then, so if you insert that gene into plant also produce antibodies. That is also a foreign body. Then automatically that animal or plant or any microorganism also produce antibodies and kill and eliminate that substance. It destroys. Then how? So there must be some protection from the antibodies. That means the host should not produce antibodies, should not eliminate this gene. That means some special gene carriers are desirable. So scientists invented that one also. Yeah. That means this is the procedure first. First they extract the desirable gene, then they produce or collect. It is also available in the market, in the laboratory. So selecting a proper vector, then clubbing these two, then producing rDNA. That combination itself is known as rDNA, recombinant DNA. Then selecting a proper host, that may be plant or animal or microorganism, selecting proper host, then inserting this rDNA, and finally developing and finally getting desirable results. This is the procedure. That means, what is the procedure in DNA technology? First, extraction or isolation of desirable gene. This is the first. Then after extracting that, then again isolation or separation or selection, whatever, of desirable vector. Second one. Then inserting this gene into the vector, that leads to formation of rDNA. Then formation of recombinant DNA, smaller DNA. This is the third step. Then fourth step, selecting a proper or desirable host. Then after selecting host, inserting of this rDNA into host, inserting or introduction or introducing rDNA into the host. Then finally getting desirable result. So these are the steps involved in DNA technology. First, they extract a specific gene. How to extract the gene? See here, to extract the gene, so number of methods are there. So first they collect some tissue. After collecting some tissue, then they dip some enzymes, enzymatic treatment. Or enzyme treatment. First selecting tissue, then enzyme treatment. By the enzyme treatment or detergent treatment, even they can use some soaps also. Then automatically all cell wall, cell membrane, nuclear envelope, they all going to eliminate. Then after enzymatic treatment, they select chromosomes. After selecting the chromosomes, they apply restriction, endonuclease. When we apply this restriction endonuclease REN, this enzyme particularly cut and separate a desirable gene. It's a gene cutter. Restriction endonuclease is a gene cutter. It cuts That's why it is known as molecular scissor, gene cutter, genetic scissor, something like that. Restriction endonuclease, which is useful to cut a specific gene. Okay, then after separation, they have to confirm it. They have to confirm it. Is it? So, to confirm that, they use Southern blotting test. Yeah. The Southern blotting test is useful to diagnose, detect, confirm the DNA. We know this one is already. Yeah. The gene cutter, this is the gene confirmative test. Yeah. So, like that, after collecting some tissue, after enzymatic treatment, they select the chromosomes, then finally, by using restriction endonuclease, they cut the specific gene, and finally they confirm by using Southern blotting test. So, this is the procedure which involved in the extracting of the gene. Otherwise, now gene computers are there. So, if you apply, simple, that means computers are there available. If you give some program, I want insulin gene, something program, what insulin, total program also already inbuilt. Just to type insulin, that's it. Automatically the system itself prepare insulin gene and it's applied. Such a gene producing machinery computer is known as gene mission. Gene mission. By using either gene missions or this is the original method. By using this method, they extract a particular gene first. Then, after extraction of the gene, they select a particular vector. Is it? What is this vector? See, this vector also a gene, is also a DNA. Present in some specific bacteria, microorganisms known as plasmid. Even scientists produce a number of artificial plasmids known as cosmids. Then, after extracting the gene, they select a plasmid that itself is known as a vector, gene carrier. Vector means carrier. Yeah. Then, after selecting this plasmid gene vector, they insert this gene into the plasmid. How to insert this vector also a DNA? To cut that, restriction endonuclease is useful. Then they cut this by using endorestriction enzyme, restriction endonuclease. Then they insert the gene between the edges. Then they paste it with the help of ligase, gene paster. Yesterday class we discussed basic. Yeah. That means here they use restriction endonuclease to cut the gene, to cut the plasmid, and finally they insert this gene, and finally they paste by using ligase, an enzyme which pastes the gene paster. So now this is known as rDNA, recombinant DNA, formation of rDNA. Yeah. Then after formation of rDNA, what to do? That question arises. To produce insulin, to produce growth hormone, to produce any medical substance, enzyme, hormone, vitamin, or any biochemical substance, they select a microorganism. That is E. coli, a microorganism. Yeah. So then after selecting a microorganism, they insert this RDNA into the E. coli. How to insert? Now number of micropipettes are available. The micropipettes also known as gene guns. Gene gun. Yeah. By using gene gun, they insert this RDNA into the microorganism E. coli. Then after inserting, this E. coli is known as genetically modified microorganism now. This is genetically modified organism, GMO. So after genetical transformation, genetical modification, then they develop these microorganisms in tissue culture. For developing microorganism, that's why that is known as microbial culture. So after collecting one trait, carbohydrates, proteins, fats, vitamins, minerals, all they mix in liquid form. Then finally they put these microorganisms into that liquid medium. Then automatically the microorganism, after observing food material, it divides into 2, 4, 4, 8, 8, 16. Then automatically within 1 week, tray full. Then again they separate into different trays. Then again 1 month, lakhs of trays full. Then within 1 year, blocks, big rooms, godowns full. So such a microbial culture is also known as cloning. Why that is known as cloning? They produce similar copies. All that microorganisms also include this RDNA. Then after this cloning process, automatically this microorganism produce a particular substance. If this gene is insulin gene, we will get here insulin. If this is a growth hormone gene, we will get here growth hormone. Is it? If this is factor 8, which is useful in hemophilia, or factor 9, hemophilia category 2B, then we produce that. That means if this is vitamin A producing gene, vitamin A, so no vitamin B producing gene, vitamin B, vitamin D producing gene, vitamin D, that may be gene, that may be enzyme or hormone or vitamin or any chemical substance or a toxic substance, whatever that one, similar substance we get. Then they extract that, purify it, then they supply into the market. And that's it. Now in market, Botox injections are available, no? How that is? Same thing, the toxin producing gene, this one, then automatically after inserting this process, is it? Finally they got that antigens, the substance. That itself is a Botox injection. If this is insulin gene, so automatically the microorganism produce insulin. That means whatever the gene we inserted, that substance we will get. Then finally after purifying, then they supply into the market. This is DNA technology. Now see, this is related to microorganism. Now we want to produce genetically modified plants, not the microorganism, not the substance. So BT cotton we want to produce. Then this is endotoxin producing gene extracted from Bacillus thuringiensis, for example, or vitamin A producing gene, then plasmid inserted into the plasmid, by using Ren and ligase, by cutting restriction endonuclease, inserting the gene and pasting by ligase, rDNA produced. Up to rDNA similar. Now, see, they collect one anther plant. Anther. What is this anther? Male reproductive part. Then the egg cell. What is this egg cell? Female egg cell. Is it? So gynoecium something. Yeah, egg cell. Then they fertilize these two artificially. That leads to formation of zygote. A single cell. Is it zygote? Now they insert this rDNA into zygote. Now this is rDNA. Same by using gene gun they insert. Then after inserting into zygote of plant cell, they develop it in tissue culture. So plants have a special capacity. A single cell itself produce abundant number of tissue. The tissue itself produce total plant, not single plant, lakhs of plants. We can separate each plant and we can plant it. Then automatically that grow. This is the procedure in botany. That means to produce transgenic plants. This is microorganism. Now we want to produce genetically modified animal. Then they collect one sperm and one ova. Then artificial fertilization that leads to formation of zygote again. Then they insert this rDNA into zygote. Then finally they insert this zygote into the mother womb. That is mother uterus, garbhashay. Then automatically the mother gives birth to that particular animal, genetically modified animal. So this is genetically modified plant. Something like that. This is totally DNA technology. So it's clear. It's clear. Yeah. DNA technology or recombinant technology, genetic engineering or genetic manipulation, everything same. DNA technology means genetic manipulation or genetic engineering is known as DNA technology. Yeah. DNA technology is a specially designed technique useful to get desirable results by manipulating DNA. There is a gene. There is a gene. That is a definition. In this process, yes. So, some steps involved here. What are that? First, isolation of a desirable gene. First step. Second step, again isolation or selection of a desirable vector. The vector also DNA present in microorganisms known as plasmid. The artificial plasmids are known as plasmids. Then next, formation of rDNA after inserting this gene into vector, that leads to formation of rDNA. Then after formation of rDNA, the researcher has to select a desirable host. That may be microorganism, that may be plant or animal or any. So selecting a proper host, then inserting this rDNA into the host, then finally getting desirable result. This is DNA technology. Yeah. So first they select the gene Is it? How they select the gene? By general method. By general method. So first they select some tissue, apply some detergents or enzymes, then test tube. After checking, automatically the envelopes, nuclear envelope, cellular envelope, that is a tissue, everything is going to destroy except chromosomes. Then they select that chromosome. After selecting chromosome, they apply restriction endonuclease. That enzyme cut and separate the gene. Then confirming the gene in by using southern blot. So over. Otherwise using computers, genetic missions. So something they produce gene. Then they select a plasmid. That means the vector. Then they insert this gene into the plasmid by using restriction endonuclease, gene cutter, and ligase, gene paste. Then that leads to formation of our DNA. Then here you have to decide again. You want to produce genetically modified plant, then anther and egg cell they select, then after fertilization they insert this rDNA into the zygote. Then developing the zygote into tissue culture, then producing number of genetically modified plants. If that is an animal, they select one sperm and ova, then artificial fertilization in test tube, then inserting that rDNA into the zygote, then inserting the zygote into the mother uterus, then automatically mother give birth to that genetically modified animal. No, they want to produce any substance. Then they insert this rDNA into a microorganism. 99% they use E. coli. Otherwise, even yeast also they use. So then by selecting the host microorganism, they insert this rDNA by using gene gun. Then they develop this E. coli in microbial culture, that is known as cloning process. After that, this E. coli genetically modified microorganism produce abundant number of substances depend upon the gene. If the gene is enzyme, it produces enzyme. If there is hormone, it produces hormone, or whatever that may be, depend upon the gene they produce. This is DNA technology, recombinant technology. Over. DNA technology in disease and medicine. DNA technology in medicine. Yeah, 12th chapter. See, DNA technology in medicine. What are the dimensions of the medicine? First of all, we are using the DNA technology to diagnose the disease. Second one, treatment. Third one, prevention. Yeah. That means first diagnosis. Second one, treatment. Third one, prevention. These are the three dimensions in medical field. So here we are using monoclonal antibodies and even DNA probes in diagnosing. In treatment, number of enzymes, hormones, vitamins, many. They are comes under therapeutic substances. Even we are using gene technology in gene therapy. In prevention, we are producing vaccines. DNA technology in medicine. So, diagnosis point of view, treatment point of view, prevention point of view. Yeah. What is this monoclonal antibodies? Nothing. First, they extract antibody producing gene. Then after extracting antibody producing gene, then same process over. They select vector, then insert that antibody producing gene by using restriction endonuclease and ligase. Yeah. Then finally they insert this rDNA into E. coli. Then this E. coli produce number of antibodies. Yeah. These antibodies either we can use in the treatment or to diagnose a particular disease. Yeah. So this is one method. In this first simply antibody producing gene they extract. Then again they select a proper vector. Is it? Then this is a plasmid. They insert the gene into the vector. Is it by using restriction endonuclease and ligase? Then this is rDNA. They insert, they select the microorganism E. coli, then insert it. Then they develop in microbial culture. Then automatically this microorganism produce these antibodies. Same to same, no? Same to same. This is one method. Yeah. See, now recently we are producing antibodies in some specific method known as hybridoma. Technology. This is also known as somatic hybridization. Hybridoma technology or somatic hybridization. First they collect one cancer cell, tuber cell, cancer cell, myeloma, myeloma. What is this myeloma cell? Myeloma cell and tumor cell, cancer cell, everything same. Yeah. Why they select that? What is the reason? What is the property of the cancer cell? Continuously goes on dividing and producing new cells continuously. Cancer means abnormal growth, uncontrolled growth. 1 cell into 2, 2, 4, 4, 8, 8, 16. Goes on. Tissue will enlarge. That itself cancer. Increasing of unwanted tissue. Is it? That is the cancer. That means, so The cell has abundant multiplying capacity, reproducing ability. Such a cell is known as cancer cell. First they select one cancer cell, then B lymphocytes. What are these B lymphocytes? These B lymphocytes only produce antibodies, antibody producing cell. So cancer cell, is it the high reproducing ability, abundant capacity to produce similar cells. Then B cell produce antibodies. That means first they select a cancer cell and antigen producing B cell, B lymphocyte. Then by using this hybridoma technology, they fuse these cells. This cell is known as hybrid cell or somatic hybrid cell or hybridoma cell, whatever. So this hybrid cell, hybridoma cell, this cell continuously divides, at the same time continuously goes on produce antibodies. That means abundant quantity of antibodies the cells produce. Then after extracting that antibodies, cleaning, then supplying it to market. That's it. Such antibodies are known as the antibodies which produced in hybridoma technology are known as monoclonal antibodies. That's it. That's it. Yeah. So by using this monoclonal antibodies, so technicians produce some slides known as titer slides. Titer slide. So that means a glass, small piece of glass, is it? A plate like, that is known as titer slide, SLID. Yeah. So here some these antibodies are fixed on the slide. Yeah. Then see, these are all useful to diagnose a particular disease. In laboratory, abundant number of different antigen fixed titer plates are available. Say for example HIV. If that HIV is in our blood, that antigens are present in microorganism, so in blood also that antigens are there. Is it? If this is HIV antibodies which work against the HIV, then automatically in our blood antigens are there, that antigen, this antibody club together. Then even after washing with the tap water, definitely the clubbing of antigen antibody you can't eliminate. After washing clearly under tap water, then they can observe under the microscope. Is it clotting is there? Yes, that's there. Confirm. The HIV microorganisms are there. Okay. No, not HIV, cholera, yeah, or typhoid. So typhoid, when there were typhoid microorganisms enter into our body, then our body produces antibodies. That antibody producing gene they extract, is it? Then that antibody producing B cell, then same they produce monoclonal antibodies. Then that antibodies are fixed here. Yeah. Then in laboratory, simply they collect the blood from the patient, then they put one drop. Then after washing under the tap, they observe is it clotting is there or not. Yes, clotting is there. Then confirm. That means these monoclonal antibodies are useful to diagnose or detect antigens. of a particular disease. If that antigen is confirmed, disease is confirmed. There is a diagnosing. That is monoclonal antibodies. DNA probes. What are these DNA probes? This is also a titer plate. But this is a DNA titer plate. That is not a DNA probe. Here, not antigens. Single thread. The DNA pieces are attached, fixed. Then, after extracting blood, simply they put one drop here, one drop here. In our blood, that microorganism is there. In that microorganism also this DNA are there. So, this DNA are double helical, but this is single thread. Always this single thread have a high attraction opposite to other thread. Then whenever, even though there is double thread, when it dropped here, automatically the single thread will attract and club together. Finish. Then they can diagnose it. Yes, clubbing is there, clotting is there. Then automatically they confirm it. That is DNA probe. That means, what is DNA probe? So these are all single threaded DNA fragments which are chemically treated or radioactive labeled. Why like that? If that is chemically treated, now laboratory available number of, so titer plates, simply after collecting blood, they put one drop, then immediately due to chemical treatment, if that antigen is there in the blood, then after clubbing, red color, then confirm red color, confirm. No clotting. So, blood also water is there, no? After wetting, no clotting, green color. That means safe. No such disease. Simple color indicates. Red color indicates presence, green color indicates absence of that particular. Such a plates. You have seen in medical, is it? Yeah. In clinical laboratories. Such a plates are known as, yes, titer plates. Here maybe monoclonal antibodies fixed or maybe DNA single threads are fixed. Whatever. These all titer plates only useful to detect the particular disease. Particularly here, DNA probe. DNA probe means, of course, chemically treated or radioactive labeled single helical DNA fragments are known as, is it? Yeah. DNA probes, which are useful to diagnose the DNA of a particular microorganism. This is DNA probe. Over. Then treatment. In treatment, therapeutic substances means drugs. Number of, in lab now, technicians produce abundant number of hormones, enzymes, vitamins, many by using this technology. What are that? The major one, somatotrophin, somatostatin, factor 8, factor 9, urochrome or urokinase, then interferons, humulin. These are all the major one. So the somatotrophin is useful to improve height. Yeah. For dwarf persons, if we inject the somatotrophin, definitely that increase the physical height. Yeah. That means somatotrophin is a growth promoter. Yeah. Somatostatin, the person is growing abnormally, then by using statin, we can control the growth. Yeah. That means is a growth controlling agent. This is growth promoting agent. First one. Yeah. Factor 8 and factor 9 both are useful to control hemophilia. But first one is useful to control hemophilia A, second one hemophilia B. Yeah. Then urokinase. This urokinase is an enzyme useful to dissolve blood clots in the blood vessels. Yeah. That means it is useful to dissolve the blood clots. That is urokinase. Yeah. Interferons are useful to cure cancers. Humulin is nothing but insulin. Useful to treat diagnosis, useful to treat diabetes, cancer, hemophilia A and B. Two different types of hemophilia, no? Hemophilia A and hemophilia B. So first one is a growth promoter, second one is growth controller. This is hemophilia A, this is hemophilia B. This is so to eliminate the blood clots in the blood vessel. This is cancer, anti-cancer. This is insulin itself, anti-diabetic, but known as humulin, human insulin, humulin. They're nothing but insulin only. Nothing but insulin only. So these are all different substances produced in DNA technology, which are useful to treat a particular disease. Then gene therapy. See, in gene therapy, there are three different types of gene therapies are there. See, in gene therapy, gene replacement. This is the first one. Then gene targeting is the second one. Then third one, gene augmentation. Of course, gene therapy is a juvenile, latest one, modern one, not developed properly, but of course, we are using gene therapy to cure some diseases. See here in gene therapy, we are using gene itself as a therapeutic substance. That means using of gene as a therapeutic substance, as a drug to cure the disease is known as gene therapy. Gene therapy means using gene itself as a drug to control the disease. That means using gene as a therapeutic substance to control a particular genetic disease. That is known as gene therapy. So this gene therapy is three types. Gene replacement. See, some disease causing gene is there. Then removing, eliminating that disease causing gene and inserting a healthy gene, normal gene. That itself is known as replace. Replace means eliminating and inserting. That's it, no? That's it. Eliminating of undesirable and inserting of desirable. That is known as gene replacement. Gene replacement. Then gene targeting. Sometimes elimination is not possible. That maybe very minute mutant gene are linked with some another. Sometimes we can't eliminate it. We can't remove it. Then scientists simply inactivate that. Inactivating of undesirable gene is known as gene targeting. So elimination and introducing, that is replacing. Simply neutralizing its function. Yes. Inactivating, that is gene targeting. Gene targeting. Then gene augmentation. Sometimes gene produce the substance but not sufficient amount. For example, insulin, we need per day at least 20 mg, 20 milligrams, but the gene produce only 1 mg, then it creates problem. So first we extract the same gene from our tissue, then multiplying the copies and inserting number of copies into the pancreas. Then if 1 is there, 1 gram, 20 are there, 20 grams. That means inserting of similar copies, inserting of many similar copies, abundant number of similar copies, based on the requirement. That is known as gene augmentation. That means gene therapy, using gene as a therapeutic substance to treat a particular genetic diseases is known as gene therapy. Gene therapy is three types. Gene replacement, that means eliminating undesirable gene and inserting the desirable healthy gene. That is one. Then inactivating undesirable, that is gene targeting. Then inserting number of similar copies. Number of similar copies. Is it? That is known as augmentation. Augmentation. Then prevention, vaccine. Vaccine. Yeah. Very simple here. Very simple. Vaccine production also. Yeah. So majority vaccines are antigens only. Antigen only. So for any disease, first they extract antigen producing gene. Say for example, hepatitis vaccine is there. That hepatitis antigen producing gene they extract. After extracting that, same to same, they select one vector. Then after selecting vector, insert the gene into the vector, then automatically that leads to formation of rDNA. Then they insert this rDNA into the microorganism. Then E. coli, that is genetically modified organism. Then in tissue culture development, they produce abundant number of antigens. Extracting these antigens and mixing with some fluids, supplying it to the market, that itself is known as vaccine. Such a vaccines are known as second generated vaccines, or toxoids, or unit vaccines, whatever. So DNA based vaccine only one is there, that is this one only. Hepatitis vaccine. First time this hepatitis vaccine was produced by Shanta Biotics of Hyderabad. That's why it is known as Shanta vaccine, Shanvac, HB, hepatitis B. So this is only the preventive. This is only the preventive. There is a vaccine. Only one vaccine, that is hepatitis B vaccine. In this method, of course, different methods, different vaccines are there. Field vaccines, live attenuated vaccines, is it? Anti-toxoid vaccines, subunit vaccines, DNA recombinant vaccines, many are there. We are discussing here only DNA recombinant based one. Only one vaccine only available at present related to human. Yeah. In animal, foot and mouth vaccine is available. That is also prepared under same. That is normal. That is non-DNA technology based one. Yeah. That is different. MMR, DPT, is it? Yeah. That is different vaccines. Yeah. What is the DPT? Diphtheria, pertussis, tetanus, DPT vaccine. For triple antigen known as diphtheria, pertussis, tetanus, DPT vaccine. BCG vaccine. For what purpose? Bacillus, calmette, Gurvin, TB. Is it? Yeah. MMR, mumps, measles, rubella, MMR vaccine. Is it? These are all different vaccines. We won't bother about this. Yeah. Yeah. Killed vaccines. Is it? Killed vaccines, live attenuated vaccines. That means they never kill the virus or bacteria. They convert into mild forms and mixing some fluid, directly giving that bacteria virus itself into the body, injecting or introducing through oral or subcutaneous or intravenous or intramuscular. Of course, different on the basis of introducing into the body. Vaccines are 4 types. Simply they give through oral vaccine. No, below the skin, subcutaneous. No, into the muscle, intramuscular. No, into the blood vessel, intravenous. Like that, that is different dimension. Yeah. So these are DNA technology based information. Yeah. So this is clear. Yeah. These are all comes under actually human genetic part. Maybe NCERT is the richest source of information. Everything you can get. Everything you will get. So if you read originally old NCERT, abundant information is available. Particularly old. NCERT books are excellent. There is no doubt at all. Mendelian is there, but Mendelian applications are not there. 100% they ask application only. DNA recombinant technology, 100% you will get. In botany also you will get. Zoology also you will get. Definitely. Anywhere you will get. This is normal. Bundle information availability, huge information available. Now we are in internet era, brother. Information scarcity is not there. Any information, bundle you will get. Then analyzing properly, keeping in the brain and producing an examination, that is the criteria. Availability of the matter, that is not the criteria. Now we are in globalization. What nonsense from one book, two books. Bundle. Which, how much deeper you want? Yeah, that much deeper is there. Yeah. Title plate, simply type it. Bundle, it comes. How many different types of title plates are there? Huh? So, vaccines, bundle information you will get. Huh? So, information is not the problem. We are now. Okay.